came
Yeah,
from.
back the way we came. I think that's the one we dropped over. The little one.
Okay, full wide.
Roger, moving on.
Come on, ship.
There might be a little something to the northeast there. Let's maybe let's see if I can tug on Argus and have a quick look.
I'll come down.
That was it.
So Box Canyon has been our first dive of this expedition, and we're coming close to the end of this dive over the next couple hours, and then we'll be in transit to Greater Farallones for our second dive later today. Sorry, Nav, can I just go over the timing again with you? So it's about 5:45.
Correct.
And you said
It'll take us 90 minutes to rise to the surface, or rise up to the surface, and then there's a 15 to 20 minute recovery evolution.
okay.
It's a steep slope.
It's a race.
The little kid in me is just really laughing at them.
Pinewood Derby.
Totally.
Can we take a look at that fish up ahead?
Sure.
And Danny, one of our people tuning in is curious about the dimensions of Box Canyon. This is William from Cannon Beach, Oregon. I don't really know that off the top of my head without measuring it on the map. I know that the feature that we're looking along that we wanted to survey was about 3 kilometers or so.
Zooming in.
Our navigator is doing some measurements for us on the fly.
Yeah, rim to rim, where we currently are, is about 2 kilometers. Looks like the narrowest point is about 1,500 meters.
Yeah,
And
we got it.
Headwaters to
He's close up.
mouth is about 7 kilometers.
Great, thanks for that nice close-up of that grenadier.
Okay, coming wide.
And then Box Canyon also has some couple different fingers, so it does kind of extend beyond our current area.
Into the abyss. Is that a flat nose?
It's a grenadier.
Grenadier?
look like real tube worms.
What's that?
These look like real tube worms.
Oh yeah, let's take a look
Could you
at.
drop the delta a bit, please?
Coming down. It's like a
Let's
divot.
take a look at what's in that little crevice.
Sulfide production there.
All right, I'll give you a bit of a zoom.
Yeah, I'm getting some more leash and we'll come around and have a look.
Tube worms.
Lamellobrachia.
What are they again?
Tube worms?
But did you know the species?
It's potentially Lamellobrachia.
Lamellobrachia. These are different than the ones we've seen so far in the dive. We've seen a lot of the feather duster worms. These are a little bit different, much longer tubes.
Yeah, they don't have a mouth. They mine sulfide and have symbiotic bacteria that convert that into energy for them.
Neat.
So that indicates that there's some sulfate coming out here?
Yeah. Or at least subsurface. Their roots can go down several meters.
Oh, wow.
That's cool.
Yeah.
They're still alive, aren't they?
Mm-hmm. You can see a couple of their plumes out. I will even hazard a species that is lumesi, but those are in the Gulf of Mexico.
Yeah. You have little rose heads, little flower.
There's a fish hiding in there. I see a fish tail, maybe a grenadier or
Never
snailfish.
seen one in the
Oh,
Gulf
I
of
see 'em.
Mexico.
Maybe a
Wow, this is cool.
Yeah, unexpected.
Oh, something.
There
Do they
he
typically
is.
burrow under sediment?
A lot of times, sort of the authogenic carbonate will sort of grow around them, and then they'll just be entombed.
Hmm. Those calcareous rings as part of its outer protection.
Yeah, they sort of grow a chitinous tube, and it sort of flares out a little bit at the tip as it grows. If you search on the internet, you should see some that are colored blue from a growth study. That's
Oh,
kind of
cool.
the famous photo of this species.
There you go, Wayne. Zoom in there.
Okay. Come wide.
very cool. Well, this is a new finding for us.
Okay, moving on. That current just pinned us against the wall.
Yeah, there are those famous photos. Unfortunately, now everybody thinks that they're blue.
They're the same species that are in Endeavour around the thermal vents? Or are they different?
Can be the same genus. I don't know about species. I'm just a geographer who worked for a biologist.
That's got to be a fossil.
Someone tuning in was interested in how biodiversity is an indicator of health and if any of the specific species we're seeing contribute to the health of our deep ocean.
Well, the area, in terms of biodiversity, the reason biodiversity can be an indicator of health is because if lots of species are able to survive in an area, it can indicate that it's healthy. And then if you start to lose species, meaning it's inhospitable and those animals can't adapt to changing conditions, then that can be an indicator that it's a less healthy environment. That's why when we explore an area, we're interested in looking at getting a kind of a baseline indication of the biodiversity. Some environments are not as biodiverse and others not because they're less healthy, just because they're different. So like in the South Pacific and in the where there's so much biodiversity in the coral areas, those are naturally high in biodiversity. Sometimes in the deep ocean, it's more homogeneous environment, less animals are able to live there, so it's just naturally lower in biodiversity. There's another whelk egg tower. We've been seeing quite a few of those on this dive. This is a great little rock feature to follow along here. Seeing a few different species of the Persingid stars, some sponges. Here's a nice shelf sponge. Can we zoom in on that stalk at the bottom right of the screen?
Speak up a bit, please.
Can we zoom in on the stalk that's just went off the bottom right of the screen?
That guy there?
That's it.
Okay, wait.
Zooming in. Got a spider on him. Or yeah,
Got some crinoids. I think they got a sample of this earlier because we weren't sure if it was part of one of the stalked sponges or the corals. Okay, that's great. Thank you.
okay, coming wide. Full wide.
Thank you.
Snailfish.
Go for that
Curious
camera.
one.
Very cute.
Ooh.
That fish had lips.
This is 2H.
2H, we have
2H.
arrived.
We have arrived.
We have arrived. 2H.
Okay.
You have arrived at your destination.
All right, we have about 15 minutes left.
Correct.
So let's move. This is an interesting feature, so let's keep moving along this contour if we can.
Roger.
Danny, approximately how much of our transect have we accomplished, or how far along are we?
Good question. I'll zoom out on the map and see. Pretty good, actually. We only have one more intermediate point we would have made until we got to the last point of our transect. So, 80% or so.
So we have at least 10 meters under us as well, for your information.
Okay.
You see down in the bubble cam, there's some, looks like there's some animals down there on the below us as well.
Okay, well, let's go down 10 meters and take a look.
Yeah, I can drop down and zig and zag.
Let's take a look at that long stick.
Can do.
Do we have some more worm tubes in there? Wow.
Let's zoom in. Partial zoom.
Yeah, tube worm.
Yeah. I think. Yeah, that must have been the stalk you were perceiving.
And there's some black coral there,
Yeah,
maybe
small.
Parantopathes again. We collected one of those earlier.
Okay. Coming wide. Go wide.
I think those tube worms are the most interesting find on this watch for me. I've never seen those in our sanctuary before.
Too close. Current pushing me in.
I'd like to learn more about them.
So you're reading an article from one of the grad students who was in my lab.
Oh, here's a black coral. This looks like Bathypathes. We haven't seen it on this watch. I'm not sure if we've seen it any other times, but let's get a look at that. two species that look fairly similar, Bathopathes and Trissopathes. This looks like Bathopathes to me. So got some nice stills, so we can. Oh, look, you can see the nice view of the base and the tentacles along the stalk there.
Are those tentacles that help keep off visitors?
Yeah, I think they call those the sweeper tentacles.
Sweepers.
Nice.
Great. Beautiful color, too.
Okay, coming wide.
Beautiful.
Nice work, Dan.
A little close.
Let's
There's
take a look at that. Some more worms.
Yeah, more tubes.
Big red.
Okay, partial zoom.
There's a lot of marine snow
Alright,
in the
slide to my right here.
column
Sediment's
here.
catching up with us.
Okay, coming wide.
Roger.
Oh. Full wide.
So I'd like to fire our last Niskin bottle before we come up. Be good if we could do that next to some corals, but we haven't been seeing any.
Some of our viewers are curious about the tube worms. We said before that they're supported by some of the sulfides seeping, but they were curious if methane could also be something that they're living on.
Sorry, did we want to trip in Niskin bottle now? Is that what?
I would like to do our last Niskin bottle before we come up, so
Yeah.
we only have a few minutes left, so we can.
Let me get out of my own dust storm here. It's raining on us from above.
If we see any corals on this wall, we could move towards them and take a Niskin there. If not, we'll just fire it.
Regarding the worms, I think we have a bit of work to do to learn more. This is a new species for us, and it's definitely something we'll be looking into to understand the conditions they're living in. I don't know that much about them.
I don't either. It's an exciting discovery.
It does sound like they can be in hydrocarbon seep regions as well.
Ha, the box canyon. Love it. Watch out for that wall. Do you want to pop the Niskin more towards the top of the cliff, or while we're down here in the...
Well, here is fine. I mean, because we have to start coming up soon, right?
Yes.
I was hoping to see a little group of corals that we could fire it near, but I'm not seeing that where we are now.
You could give it another 10 minutes.
Okay, let's do that. So
come back up towards the top of the cliff there. We're more likely to find them at the where it was hanging over.
And why do we want to collect water near corals?
Well, we're collecting water for two reasons, and one is to look at the eDNA. So if we can have visuals of a coral species and then collect that eDNA, that helps us. We don't know that the DNA in the water came exactly from those corals, but it does give us an indication if we see the eDNA in that sample or not. It can help us understand how well we're matching the eDNA to what we're seeing. The other reason we're collecting water is to look at the carbonate chemistry. And we're interested in how, what kind of carbonate chemistry conditions the corals are experiencing. So that can give an indication of what those corals that we're seeing are experiencing. Oh, there's something to look at.
Zooming in.
This looks like Calagorgia. We have seen this on this dive, but not a lot. It's a primnode coral. And it's just out here by itself.
Can you hit one on bubble cam?
And hi to all of our new viewers. You're catching us as we are wrapping up our Bucks Canyon dive. We should be ascending soonish, so continue enjoying these awesome slopes and deep ocean bottoms.
We can get our Niskin here if this is a good spot.
Yeah, copy that. Just trying to stabilize here.
Okay, Niskin.
And that'll be bottle 11.
Without dust in it.
Nope.
We're still moving, are we?
Don't do that.
Huh?
Don't, no, I don't.
No, I'm just going to use my stick lock.
Okay. You can go auto now if you want.
Oh, he has to switch it.
I only
He
got
hasn't
3
switched
beams.
it yet. To the camera. Okay, I got you. We'll go back to that.
Ah. That's exactly what I did not want to do.
So,
Jeez.
You're continuing to see a lot of that marine snow, which is the nutrients, the plankton that is helping to feed a lot of these deep ocean corals and other animals like the brittle stars that are all over the ocean bottom that you're seeing right
What's
now.
our time, Miles?
More tube worms.
We're real close.
Yeah.
If you want to go back to that coral or...
No, it's completely dusted. Not good. Niskin area. I was pushing in without a heading in it. Kicked around so that the lats kicked in. Just destroyed it. It's settled out. I'll go have a look.
These little white things are like kind of swimming around in front of the camera. I wonder what they are. See them?
Yeah.
Kind of darting around.
I'm not sure what they are. Could be little copepods or shrimp or something. They don't look too shrimp-like, but yeah.
okay, partial zoom.
Roger. So I want to pick up that rock. Okay, I got 4 beams there. You think it'll hold?
Yeah.
One way to find out. You're gonna move again?
Hmm?
Oh, you're moving that way.
Yeah.
Okay. It's not holding.
Okay, coming wide.
Yeah, we're gonna try and see if the vehicle will hold, and we'll pop a Niskin.
Okay.
You guys happy to pop one here?
That's great.
Stay, Simba. Okay.
Do you want to be closer?
I can, yeah. Let's get the minute out. What's our last Niskin?
11.
11. Wayne, can you iris up a little for me, please? Thank you.
You know, camera rack in.
Should be. No more racky.
Roger.
That's full iris there.
Is it?
Yeah.
It's missing a light.
Is porch still an option? Nope.
Right. Snaggletooth will get a finger on it here sooner or later. Got it there, yeah? Where's our victim go?
Right behind the arm. Yeah, you see it.
See it. So the idea is to be over the coral, is it?
Yeah, we don't have to be right on top of it, but close by.
Trying to put the Niskin close by it there. Okay. Here we go. Pulling.
Trip.
That was it?
Yep.
Here's your money.
Is that sample 22?
Yes, sample 22. Okay.
Alright, sad to say, that
What?
concludes the dive.
Well done, everyone.
Yeah, that was great.
Full
Thank
of
you.
samples.
So
Lots learned.
you can
Yes.
start coming up on Argus.
You want to get, you want to spin around and come to the south of Argus.
Roger.
Partial zoom while we wait. This is that guy again. I
You can
remember
start
him.
coming up. I'll come around once you drag me.
Bye-bye. Coming wide. Full wide.
That's Argus dragging Herc off the seabed. I
Sorry.
have to go now, sorry.
All right.
Farewell, feathery coral friend.
I in the sky is saying, come home.
All right. Bye. Bye, buddy. Hope you find your dad.
It's been Fun exploring Box Canyon.
I'm gonna be down about there-ish.
What's that?
So do you want to be down about there?
Yeah, we got 1,700 meters to work that out.
So we're finishing our first dive of the cruise in Cordell Bank National Marine Sanctuary in Box Canyon. It'll take us about 90 minutes to get to the surface, and then another 15 minutes to recover the vehicle on deck.
If you step the ship north 20, 30 meters.
Can do.
Easier than me battling it.
Does the ascent take longer than the descent? Because I guess more weight from all the samples we collected or any other factors, or are they about the same amount of time?
It's slightly slower to ascend, mainly because the starboard vertical is thrusting directly down onto that starboard bio box, and so it slows us down a bit. We don't get, you know, fully efficient thrust like we do during descent.
Sure, that makes sense.
We need an aft vert. We should stick the spare on there. Just run it out.
And Taylor Ann, some of our viewers are curious about the samples, how long we keep them, what we do with them, where do they end up. Can you share any of your knowledge around the samples that we've collected and the process around them?
Yeah, sure. So after we get back up with all of our samples, we will have people from the wet lab come out and retrieve them from the ROV and bring them back to the lab and store them in a fridge to keep them at a constant temperature. And then we will process each of those samples by placing them on like a mat with a ruler for reference of size. And we will take note of what species we think they are, take samples to process for like their DNA to actually get a genomic understanding of what organism we're looking at. And I'm not too sure how identifying will go. I haven't done it before, but I'm looking forward to taking part in that process. And then you can tell them a little bit about where they get sent off to after we're done processing them in the lab.
Sure, so we have a number of partners that are interested in these samples depending on what we've collected. So they go off to many different institutions for further investigation. And then they're all housed eventually at the biological, all the biological samples, excuse me, are housed at Harvard University's Museum of Comparative Zoology.
And this is Danny. Because we're in Cordell Bank National Marine Sanctuary and all the specimens that have been collected in the sanctuary are archived at California Academy of Sciences, and we have Gary Williams on board, who's a coral expert. We send all our invertebrate samples in duplicate, so we do send the samples to Harvard, but we send the duplicates and the larger pieces of the sample to Cal Academy, where they get archived. And Gary will look at all the coral specimens, and if he doesn't have somebody in-house to look at some of the other species, they will work hard to find somebody who is an expert in that and can identify them. For the core samples, sediment samples that we've taken, we freeze those samples whole, and they will be analyzed by scientists at Bodega Marine Lab. They're looking at the microfossils in the sediment to look at the animals that have lived here historically. And then the water samples we're looking at for the eDNA, so that the water gets filtered out and the materials in the water ends up on a filter that can then be, DNA can be extracted from that and analyzed. We're also doing using the water to look at carbonate chemistry, so that water gets placed into bottles and then preserved so that it can be analyzed back at the lab. The chemical content of the water can be analyzed back in the lab.
Great, so the learnings continue to unfold when we bring all these samples up from the deep ocean.
Right, that's a good point, that the surveys that we do while we're here on the boat are just a really small portion of the study. It's just the beginning of the study, actually, because we will be looking at the video back in the lab, the data that the vehicles have recorded, the samples that we take back. So we generate a lot of information that, and the study continues from here. It'll probably take us a year or so from the end of the cruise to analyze all the data from this cruise.
Lots of work to be done.
And then at the end, we have a great characterization of what these habitats that we're exploring are all about. So we know more about what things we can do to best protect them, what new questions come up. I know new questions have come up. Now we have a new species that we need to learn about, these tube worms, which is pretty cool.
And this is our second time on Nautilus. Our first time was back in 2017, so this is only our second time looking at this really deep habitat. But we have done other ROV cruises looking at the seafloor in the sanctuary in shallower areas, and you can read a little bit about that on our website, cordellbank.noaa.gov. And for example, at the Cordell Bank itself, which is the feature that the sanctuary was originally designated to protect and has been focused around, and there have been scuba surveys. This is back in the 70s and 80s, and submersible surveys in the early 2000s. And then some ROV surveys. Most recently, we were visiting Cordell Bank in 2014, 2017, and 2018. So we have some historic data that we're now looking to look at, and to be able to because it's now a well-characterized habitat, to be able to monitor that feature over time and see how it might be changing.
We know the deep water environment doesn't change as quickly as the surface water environment does. You know, the surface waters have temperature fluctuations, oxygen fluctuations, chemistry fluctuations, so much more changing rapidly, whereas the deep sea remains relatively stable. It takes a long time to shift the deep sea.
We have another research project that we've been doing for 16 years. It's called Applied California Current Ecosystem Studies, and this is a collaborative project that our co-lead on this cruise, Jan Roletto, and I work on together with Point Blue Conservation Science. And it's a project that looks at the pelagic environment, looking at the surface of the ocean for the predators, the seabirds and marine mammals, the prey that they consume, the krill and the fish, and then the ocean conditions that support that. So we have 16 years of data looking at the surface and the down to 200 meters of the prey that those animals target. And so we can then kind of be thinking about what do we see at the surface and what are we seeing at the bottom of the ocean. And there are some linkages there. So this food that the animals at the bottom of the ocean consume comes from the surface. So those changes that are happening at the surface could be the linkage that affects the deep sea.
It'll be exciting to hear Karina's results at Bodega Marine Lab about studying bamboo corals to understand that. I'm just fascinated by the fact that there's this deep sea coral that can record information that tells us more about the surface waters, and I'm just so curious how we found that out in science. It's all connected. Let's pull up our oxygen and temperature and salinity, see what that looks like.
That's a good idea, because as we come out through the water column, we will be seeing some changes. We've been seeing really constant temperatures and conditions while we've been exploring the seafloor. About 2 degrees Celsius, 2.25 or so, but we'll start to see it come up. It'll be interesting to see how quickly that comes up as we move through the water column.
I think the sun is just starting to come up outside.
Beautiful.
Or will be.
So our plan is to have the ROV on deck by 8. It'll take us a few hours to process all the samples that we've collected, and then for the ROV team to get the vehicle ready for the next dive. The plan is to move the vessel up north and to Greater Farallones National Marine Sanctuary and do a dive there. So we're sharing this cruise, this joint cruise between Cordell Bank and Greater Farallones National Marine Sanctuary. So we're trying to split our time evenly, make sure that each sanctuary has a chance to explore and reach their objectives.
We call these ascents the, blue water time. And often this is when we can see some unexpected visitors. We've seen a sperm whale most notably once when on an ascent or a descent in the blue water zone. Some of our viewers were curious about the potential to see gray whales right now.
I can talk about that a little bit.
Thank
so
you.
gray whales migrate really close to the coast, and they go from Alaska to Baja in the winter months, usually between December and May. And we don't see them too far offshore often, although there are a couple that hang out around the Farallon Islands themselves year-round. The food's so good, they don't migrate north. But let's see, so it's October, probably not that likely to see grays, not impossible. There are a couple that hang around year-round in this area. We still have the opportunity to see humpback whales and blue whales and fin whales offshore here, as well as Rizzo's dolphins, Dahl's porpoise, Pacific white-sided dolphin, northern right-whale dolphins, and pilot whales, pretty rare. Really interesting find with the Greater Farallones National Marine Sanctuary Beach Watch program that we were talking about on our last watch. These are citizen scientists that get training with the sanctuary, the Greater Farallones Sanctuary, and they monitor the beaches throughout the sanctuary area. And they are really the eyes and the ears of what washes up on the beaches. And about 2 months ago, a Beach Watch volunteer found a neonatal beaked whale. And I can't remember the name of it, but it was pretty cool because beaked whales are so rare. We hardly ever see them because they're so elusive and they live in such deep water areas. So it was a pretty interesting find for those Beach Watch volunteers.
That's very cool, and just speaks to the power of community scientists. When you have more eyes and ears out and hands participating in research, often you're able to collect more data points and actually have more awareness for some of these activities and see things that scientists wouldn't otherwise be able to catch. So, community scientists, way to go.
Yeah, I think they were pretty excited. The two volunteers that found this specimen. We're veteran beach watch volunteers, and we're pretty excited because most of the time it's some dead birds, maybe a dead sea lion here and there. But it's pretty great. They get to visit a beach, I believe, once or twice a month, and they really get to know that beach over time, and its seasonal changes, the things that wash up seasonally, some of them natural, some of them unnatural, and really become a great treasure trove to help the sanctuary know what's going on, what's washing up on the beach, and what is that indicative of. They're also legal guardians. I guess that's not the right word, but they are able to take samples of oil if oil wash up on the beach. They are a legal custody, meaning they're certified to take a sample that can be used in legal ways, and they can fingerprint the oil to a ship, to a source, and it's pretty important when we have these incredible beaches, these incredible habitats right outside the Golden Gate where ships come and go all day long. It's a major shipping port and shipping lanes coming in and out. There's just a lot of potential for accidents to happen, and there have been a couple. So good work, Beach Watch folks. Ooh, I just saw a glimpse of the sunrise. That was pretty.
Yeah, so other ways that we spend our time when we're not in the control room is with binoculars in hand and keeping an eye out for some of this megafauna, both marine mammals, but also marine birds that are in the area. And that's a really cool way to pass a few hours as well.
Yes.